Okay, it's 2 p.m. in Singapore on Thursday, 4th of March, 2021. So let's get started. First of all, on behalf of the SingHealth Duke-NUS Global Health Institute, I'm delighted to welcome all of you to this webinar. The talk today is part of our monthly Global Health Seminar series, during which we present a range of pertinent global health topics and engage our community in Singapore and beyond. Today's topic covers the science of COVID-19 vaccines and considers what is needed to make our world safer, obviously something that touches all our lives. Our audience today reflects that interest because we have over 400 people registered for the talk, and they're logging in from countries across the region. So we're really delighted to have all of you with us in the room. And it's no wonder we have this level of interest because we have with us two of the leading Singapore experts in this field. We have Professor Ooi Eng Eong and Professor Jenny Lowe. And we'll do a quick introduction of them, but it certainly doesn't cover all their accomplishments because I'll take up half our time, so we're not going to do that. But I will just briefly say, together, they're the co-directors of the Viral Research and Experimental Medicine Center at the SingHealth Duke-NUS Academic Medical Center. Their center aims to develop molecular endpoints for use in viral disease therapeutics and vaccine trials. And this certainly provided a platform for the development of a vaccine candidate that has been co-developed by them. And hopefully we'll be hearing more about that soon. So now a little bit about each of them. Professor Ooi trained in medicine at the University of Nottingham and then completed his PhD studies at the Department of Microbiology at the National University of Singapore. He holds concurrent appointments with the program in emerging infectious diseases at Duke-NUS, the Sorsvik Hawk School of Public Health, and the Department of Microbiology and Immunology at the Yong Loo School of Medicine. He is also faculty of our Global Health Institute. Amongst his numerous awards, he has received the Clinician Scientist Award by the National Medical Research Council of Singapore in 2010, 2014, and again in 2019. So as I said, there's so much more we could say, but I'll move to Professor Jenny Lowe. She's a board-certified senior consultant with the Department of Infectious Diseases in Singapore General Hospital, an associate professor with the program in emerging infectious diseases at Duke-NUS. Her current research focus is on early phase adaptive clinical trials of viral therapeutics and vaccine development, as well as the role of innate immune responses in modulating the outcome of infection or vaccination. She was twice awarded the National Clinician Scientist Award in 2016 and in 2019. So it's a great honor to have them with us here today, and I'm really looking forward to their presentation and our conversation with them. Before I hand over, a few housekeeping notes. Please keep yourself on mute. The webinar is being recorded, and you can post your questions via the chat box at any time during the presentation. They will be addressed at the end. So over to you, Eng Eong.
Thank you very much, Amina, and thanks for a very kind introduction. So what we thought we'd do today is basically maybe just frame some things for us to discuss, and obviously very topical given what's happening right now in the world. And then maybe we'll go on to have some discussions on vaccines and issues of vaccines, if any, and how do we best manage that. So I'll start, and then I'll hand over the time to Jenny. So probably we don't need an introduction to this map. We know what's happening in the world today. So this is, I took this, I think as of yesterday or 2 days ago from the WHO website. And this map basically shows you where the deaths are. And the darker the color, the greater the rates of deaths from COVID-19. So right now there are over 100 million cases of COVID-19 in the world. And well over 2.5 million deaths by now. So, and this number is set to continue to grow. And part of the reason, and all that happened over the course of just slightly over a year, and that shows you the magnitude and efficiency of how this virus spreads across the world. And because of the severe disease and the very poor outcome, especially in older adults and adults with chronic diseases, that has fueled a lot of development, both in terms of drugs and vaccines. And obviously vaccines then attract a lot of attention because it can prevent us from getting COVID-19. And so this is the state of vaccine development today. So slightly over 100 vaccines at various stages of clinical trials and approval. So in the world right now, in various parts, if you add all of those that have received approval, there are 6. Some have received a limited authorization for use, and there are about almost 100 in clinical trials right now. And the one that we're developing, at least in partnership with Actaris, is right here in phase 2. We hope to progress to phase 3 pretty soon. 4 have been stopped because of various issues. And the early indicators, I mean, vaccination rolled out at the end of December, and we're in early March right now, and there are already indicators that it's having an effect. And this data from Israel, and I'll show you why we're using Israel, but they have a very high vaccination rate. And you can see that those who received the vaccines early are now having a lot fewer hospitalization than those who received the vaccines late. Suggesting that the vaccine has the effect of preventing disease, severe disease, and death, and so that bodes very well for our effort to control COVID and hopefully return our lives back to as much of what was normal in the past as possible. And this is I'm sure some of you would have seen this graphic. It was taken from the Straits Times just earlier this week. And this is the vaccination rate in different countries. So this rate just shows you the proportion of the population that has received at least 1 dose of the vaccine. And Singapore is right here. And so this is so about 6% of the population has received 1 dose. Fewer than 5% received 2 doses. So Israel, of course, is leading the pack, and then followed by UAE and some other countries in Europe. So what you see here could, has a number of implications. One is the commitment of the leadership of these countries towards vaccination. But 2, and possibly also, and there's a third reason, 2, the willingness of these governments to pay for vaccines. So one of the reasons why some countries are getting more vaccines than others, because they're willing to pay more. And thirdly, we all know that there's some issues with how many of us would accept these vaccines. And so these are data from some surveys done in the various countries. So vaccination rates can be as high as in the 80s or down into less than 50% depending on where you are in the world, right? And so that presents some problem. And we've all heard about vaccine hesitancy. I'm not going to talk about it, but suffice to say that I'm going to show you a series of slides to kind of show you that a lot of things that's happening to us now is actually not new, right? So this is smallpox, and instead of showing you a graphic picture of someone with smallpox, I'll show you a mummified person with smallpox, because we have just finished lunch, most of us, and it's not very appetizing to go and see someone with smallpox. Right? So this is actually an Egyptian mummy, and you can see that there are pock marks on these on the face of this mummy. And smallpox has been around for a very long time, until, of course, in about 1970 when it was declared extinct. And the history of smallpox, we probably know, we successfully got rid of it because of a vaccine. And the reason and the motivation for a vaccine, or the discovery of a vaccine, was actually made because of observations that even lay people have noticed, right? So this is a 18th century rhyme. I'm sure many of you would know this. So it says, "Where are you going, my pretty maid? I'm going a milking, sir." She said, blah, blah, blah. And in the interest of time, I'll cut to the last part. "What is your fortune, my pretty maid? My face is my fortune, sir," she said. And the reason why it's called pretty is because they don't get pock marks on their face, right? Instead, they get them on their hands. And so that was how Edward Jenner came to think that this, whatever that he could find here, would protect people from smallpox. So if you were to grow up at that time in Europe and even Asia, many of us, by the time, many of people who would have grown up to become adults would have pock marks on their face because they would have acquired smallpox as children. Except these maids who spend a day milking cows, they get their pock marks on their hands instead. And so Jenner therefore reasoned that whatever is in here is going to protect people from smallpox, and he did this experiment, as we now know, on a neighbor's child, James Phipps, where he inoculated first the cowpox, which was extracted from the hand of Sarah Nelms, into this child, and then after that followed by smallpox. And happily, James Phipps survived and did not get smallpox. So actually, although Edward Jenner discovered or showed for the first time that there's this thing called a vaccine, he never actually used the word vaccine. The term vaccine came much later, and it was actually coined by Louis Pasteur when he developed a vaccine against fowl cholera bacteria. And then he named it vaccine in honor of Jenner, because vacca is the Latin word for cow, right? But for scientists among us in this audience, this is the encouragement that we sometimes all need, right? Even Jenner had difficulty publishing his paper, right? That his reviewers' comments were that this was in variance with established knowledge, and he had better not promulgate such a wild idea if he valued his reputation. It was so bad that Jenner had to publish this at his own expense by basically making it into a pamphlet, and but the information here then became how we develop vaccines. And then for those who are working on vaccine, once in a while you get you get called names, right? So now this is Lord Byron's response to the vaccine. So now look around and turn each trifling page. Survey the previous works that pleased the age. What varied wonders tempt us as they pass. The cowpox, tractors, galvanism, and gas in turn appears, right? And this appeared in the popular press at that time. And this is a collection from the Wellcome Trust's library, where you see these people are now spouting cows out of their hands, their mouths, horns on their heads and all that. So people are making fun of cowpox, right? It's not unlike what we are facing today, that we face concerns or rumors that RNA vaccine will somehow change our DNA and change our well-being, right? But again, the lesson for us is that we should press on and trust the science, right? And this was eventually what the recognition that was bestowed on Jenner, right? Even a letter from President Thomas Jefferson telling him, that what he has done will never be forgotten. And Napoleon, right, even who was fighting a war with English, said he could not refuse anything to such a great benefactor of mankind. So whatever that we are facing with vaccines, even when it's new, and the fear of something that's new and the unknown is not new. But let me maybe now switch gear a bit and show you where the state of play is. So there are different kinds of vaccines, right? So the idea is that for those who are non-scientists in this audience, this is a schematic of a virus. It's a very simple organism. All it has is a genetic material, either in the form of DNA or RNA. And in SARS coronavirus 2, this is in the form of RNA, never both. So it's either DNA or RNA. In our bodies, we can find DNA and RNA, but not in the virus. You only find one. And then it's surrounded by a protein coat. And that's all there is to it. Right? So whereas this thing, when it gets into cells, then can cause disease. So one way of making a vaccine, therefore, is to weaken this virus, and sometimes called the live attenuated vaccine. So it can still cause an infection, it just doesn't cause disease. And this is arguably the best form of vaccine in the world, right? And this is the one kind of vaccine that can give you potentially lifelong protection with one single shot, because it simulates an infection. It actually causes an infection, but no disease. But such a vaccine is hard to make. So therefore, people then, as a rapid response, turn to these other forms of vaccine. So some use chemicals to kill the virus. You grow it up and then you use chemical to kill the virus, or you put the spike gene into another virus. In this case, mostly adenovirus. So that's what AstraZeneca, Johnson & Johnson, and the Russians did. Or you can also just express the spike protein on its own and then use that as a vaccine. And that's what Novavax has done. Or you can use the genetic material, but not of the whole virus, only in those parts that you want for the immune system to recognize and develop memory against. And that's what Pfizer and Moderna has done with RNA vaccine. And that's what we're doing with Arcturus, with a self-replicating RNA vaccine. I'm not going to go into the details of the vaccine, but I want to show you that it actually works, right? And this is the phase 3 data from the Pfizer BioNTech vaccine that we're using in Singapore. And the same trend is seen for Moderna's vaccine, the other one that's licensed for use in Singapore. And this is Israel's real world data. So this is clinical trial data, this is real world data, and I'll take you through both. So this vaccine is given as a 2-dose vaccine, one at the start, and then 21 days later, a second dose is given, right? And then, of course, they say that the efficacy measured at this point shows a 95% efficacy. Okay, but if you look at this line, it flattened out just before day 14. In fact, the clinical trial people realized this and blew this part up, right? And you see the line diverge around about day 11, day 12, it started to flatten out, whereas the placebo continued to rise. And this is the real world data in Israel, where this is, it's not a trial. This is actually rolling it out to the population, and over 1 million people, that's what they saw as well, right? That this is the placebo line and this is the vaccine line. And again, the divergence occurred at around day 12, right? And this is for all COVID cases and this is for severe COVID cases. So that tells you that something must be happening at this point, right? So it tracks along nicely with placebo until something then happened here that then allowed the immune system to now protect people who are vaccinated, whereas if you're not vaccinated, you continue to get disease. So now in the earlier studies, what they did was measure the immune response at day 21 and then again after the second dose, right? But what happened at this early time point, no one knew. And that's what we did very rapidly with, in collaboration with Jenny, with Shirin Kalimuddin, with Jean, all from the Singapore General Hospital ID department, as well as Antonio Bertoletti, who did a lot of the T cells. And so what did we find? We found that, and this is in the first 20 cases, first 20 people who received the vaccine in Singapore. So what we found was that around about day 10, so if you think about it, the incubation period of SARS-CoV-2 infection is about 2 to 5 days, right? So you get infected 2 or 5 days later, then you start to get COVID. So whatever protected you from COVID must happen about 2 to 5 days before. And that's why we're interested in this period in time, about day 10 after vaccination. So at this point in time, 80% of people have antibodies in the form of IgG after vaccination. And at day 10, they also have IgA, also another form of antibody. In other words, you get decent levels of antibody, but these antibodies do not stop the virus from binding the ACE2 receptor, which it needs to get into the cell. Neither does it neutralize the virus. And yet, or at least most of them, right? Only 20% of them have such antibodies here and only 15% of neutralizing antibodies. And yet the world is very focused on trying to say that if you do not have neutralizing antibodies, there will be no protection. That's not true, right? As long as you have all total antibodies seems to protect, and you have T cells. And that's the remarkable thing about RNA vaccine. You get pretty good T cell response. And this is measured by an assay that Antonio Bertoletti's lab has developed, that all of the people at day 7 and 10, you can find T cell responses. And this is a more conventional assay where, again, we find a majority have T cell response around about day 10, right? So that would suggest that antibodies and T cells, at least in the Pfizer's vaccine, could contribute to protecting against COVID-19 without neutralizing antibodies. It doesn't mean that neutralizing antibodies won't protect. It just means that it's not absolutely needed, right? So this idea that T cells could protect, right? Is there any evidence for it? Let me start off with an experiment that our lab did, and then I'll hand over the time to Jenny for the more clinical evidence. And so this is a vaccine that we are developing in Singapore in collaboration with Arcturus. So it's called LUNA-CoV-19, and many of you might have read it in the newspapers. And so what we did in this experiment is that we gave mouse the vaccine, and then after 30 days later, we challenged them with the virus, right? I'll skip this because it doesn't matter so much. So if you do not vaccinate, so this is the group of mice that got placebo, you see this, very remarkable and very rapid weight loss. And there's the clinical score. So they were getting sick very rapidly. And then they died, all of them, by day 7. But if you vaccinate the mice and then you challenge, then all of them survive, no weight loss, no clinical sign, and 100% survival. So all good, right? But this vaccine is an RNA vaccine which produces both antibody and T cells. The question is, which one is more important? Okay, so what we did in this experiment is we either get rid of the antibody producing cells or we get rid of the T cells, okay, or both, right? And this is the control. So if I get rid of the critical one, I will lose the protection, right? So the control are both protected in this infection in the lung and the brain, right? If you get rid of the antibody producing cells, you still are protected. You get rid of the T cells now, and you lose the protection. Just as if you get rid of both antibody producing and the T cells, then again, you lose the protection, but not any more than if you just remove the T cells. So that suggests that T cells play a very important role, at least in the in the protection that will be conferred by an RNA vaccine, right? So I'll hand the time over to Jenny, who will tell you about the clinical evidence for protective role of T cells.
Okay, thanks, Eng Eong. Yeah, so I'm now going to move into the clinical observation studies to illustrate the importance of T cell immunity against acute viral infections. And I'll start off with this paper that was published a couple of years ago by, so Eng Eong at that time, together with the NUH pediatric colleagues, described a case of persistent dengue infection in a renal transplant patient. So I'm going to talk a little bit about this case because this is dengue, but I think that was around that time, it also really made, through this case example, made us appreciate the importance of T cell immunity in a control of viral infections like dengue at that time. Okay, so this patient, she has systemic lupus erythematosus SLE, which is the short form that all clinicians use for this condition. And she has SLE since she was a teenager. And when she was 23 years old, because of severe lupus nephritis causing end-stage renal failure, she underwent unrelated living donor kidney transplant. And unfortunately, post-transplant, she suffered acute graft versus host disease. And the nephrologist had to give her high-dose anti-thymocyte globulin to manage the graft versus host disease, which actually made her even more immunosuppressed as a result. And she was maintained on prednisolone and tacrolimus with mycophenolate post-transplant. And about 3 months after transplant, she developed fever with body aches, diarrhea, and was eventually diagnosed to have acute dengue 3 infection through both PCR and NS1 tests. And during this period, because she was considered very immunosuppressed, the MMF was removed, and because they wanted to reduce the pressure of the immunosuppression and wanted to aid the recovery of the dengue infection. Then she was just maintained on prednisolone and tacrolimus. And what was interesting subsequently was, despite her recovering from the acute dengue, so she had, fortunately for her, she had quite an uneventful clinical course. Her fever resolved within a few days, and then she had platelet recovery, and she became clinically asymptomatic. But the nephrologist kept testing her blood and found that she was persistently positive for dengue 3 via PCR, even up to 4 months post-infection, as you can see from the graph there. And they continue to be able to isolate the virus, the dengue 3 virus, from the urine until up to 9 months post dengue infection. And in fact, the dengue viruria, so this is from the urine, didn't disappear until her CD4 count went above the lower limit of normal. So this persistence of the dengue virus in the blood and urine, despite her having detectable antibodies, you can see that actually her antibody to dengue 3 was very high. And that, the fact that she only fully resolved the dengue infection by having a negative urine PCR result, that coincided with the CD8 count, suggests that actually the cellular immunity played a very critical role in sterilizing the dengue infection in this patient. Okay, so now I'm going to show you the rest of the clinical evidence, which are all COVID related. Some of you may have seen this paper. This is a Nature paper that was published sometime last year. So at the start of COVID outbreak in Singapore, we thought that it was there was a interest at that time to try to understand whether the ex-SARS patients in Singapore would have any evidence of protection against SARS-CoV-2 infection. And if you remember in 2003, when we had the outbreak in Singapore, many of the SARS patients were healthcare workers. And so with IRB approval and by word of mouth, we managed to recruit 15 of these SARS survivors to do this particular study. So when we did the study last year, they were 17 years post SARS infection. And again, these works were all done in Antonio's lab. We tested to see whether these healthcare workers have any evidence of T cells against SARS-CoV-1 as well as SARS-CoV-2. So what Antonio found in his lab was that these individuals not only have gamma interferon response to the SARS-CoV-1 peptides in their T cells, but they were also able to expand these T cells when he stimulated the T cells ex vivo using the nucleoprotein peptides of SARS-CoV-2. So this paper illustrated that virus-specific T cells induced by infection by other beta coronaviruses can be very long-lasting. In this case, up to 17 years, and that patients with COVID-19 will develop long-term, suggests that it is very possible that patients with COVID-19 will develop long-term T-cell immunity after infection. And even cross-reactive T-cells from other beta coronaviruses may potentially protect or modify the pathology caused by SARS-CoV-2. Okay. Next slide, Eong. So then as the pandemic sort of evolved in 2020, we were then able to longitudinally follow up a subgroup of COVID-19 patients. And this paper was published in Cell Reports. You can see that, again, this is collaboration with Antonio's group, NCID, and various hospitals. What we did, this was the number of patients follow up in this particular report is a small number of patients. So there were 12 patients that were follow up from symptom onset to either convalescence, meaning they recovered, total recovery, or death. And we quantified, because we were tracking them prospectively, we were able to quantify the SARS-CoV-2 viral load in the upper respiratory tract through throat swab. And the SARS-CoV-2 specific antibodies, as well as the T cells at multiple time points from acute infection all the way through convalescence or until death. And as you can see from the various graphs there, there's a direct association between early induction of the functional SARS-CoV-2 specific T cells and rapid control of the viral infections. And in fact, early induction of the functional SARS-CoV-2 specific T cells was observed in patients with mild disease and rapid viral clearance compared to those with more severe disease. So again, this paper again, suggested that T cells play a very critical role in the control and resolution of SARS-CoV-2. Okay, then I'll move on to this paper that just came out yesterday, in fact. You remember that at the height of the outbreak in 2020 in Singapore, there were many clusters of cases among foreign workers who are they live in these huge and very densely populated dormitories, either in thousands or sometimes 10 over 1,000 workers live in one such dormitory. So when the outbreaks happen in these dormitories, we will then, that kind of gave us an opportunity for us to follow up these workers prospectively. So this collaboration, again, a lot of people were involved. Social Hawk, together with Antonio's lab, and then the various hospitals, mainly, I think, let me see, NUH, NCID, SGH were involved in this collaboration. For about almost 500 dormitory workers were followed up longitudinally, prospectively during that period. And 85 individuals had detailed T cells analysis done. And we then use this 85 individuals data and to compare it with another 75 patients who were proven to have SARS-CoV-2, and who were hospitalized. And these 75 symptomatic patients had a range of clinical presentations. So some of them had mild disease and some moderate to severe. So the 85 asymptomatic foreign workers were compared with the 75 symptomatic patients who were hospitalized. Just some key findings from this paper. What we found was that when we compared the SARS-CoV-2 specific T cell response of the asymptomatic cohort with the hospitalized cohort, actually the overall magnitude of T cell responses against the different spike proteins were similar in both asymptomatic and symptomatic cohort. What's very interesting is the kind of cytokines that the T cells produce were different. So if you look at the figures below, the T cells that were induced by in the asymptomatic group, they secrete more gamma interferon and IL-2, which is more Th1. And it seems that they have a more coordinated production of the pro-inflammatory and regulatory cytokines compared to symptomatic patients. So this paper is important because some people, earlier on, some people thought that in asymptomatic in asymptomatic SARS-CoV-2 infection, there may be a weaker immune response. So this finding actually suggests that clearing the virus, being able to clear the virus without developing symptoms is actually not associated with a weaker immune response. And in fact, well, on the contrary, there is quite a robust and well-coordinated virus-specific T cell response, which I suppose in combination with antibody production actually protect these people from developing symptomatic disease. Okay, next slide, Eong. Thanks. Okay, so this study is not out in peer-reviewed journal yet. It was posted in bioRxiv towards the end of last year. It's a study that was conducted in UK. This group recruited more than 2,800 participants, either working in the hospitals or in the fire and police services throughout England. And the there was about 2,600 volunteers recruited, irrespective of their initial PCR, SARS-CoV-2 PCR results. Then they had a small cohort of 154 subjects who were recruited based on known positive PCR results. The study started in June, in the middle of last year, and at recruitment, they measured all the volunteers' interferon, the gamma interferon secreting function using the T-spot. And they measured antibodies as well using commercial assays. Then they described the time to microbiologically confirmed SARS-CoV-2 infection. Then they stratified these cases by the various immunological parameters. So what you can see here is very interesting that they derive a cutoff of a T-spot count of 12 counts, and individuals who have high T-cell response, meaning they have more than 12 spots on the T-spot assay, about half of them had detectable antibodies. The other half actually were negative for antibodies. And none of them developed or came down with COVID in the 3 months of follow-up after June. So these individuals were followed up to about September. But in the group with T-cell count, with low T-cell count defined as T-spot less than 12 spots, there were 20 individuals that developed COVID-19 during that period. So this group came to the conclusion that peripheral blood SARS-CoV-2 responsive T-cell numbers actually associated with the risk of developing COVID-19. Okay, then we'll move on to this last paper that I think that just came out just 1 week ago. I think it came out in bioRxiv last week. This study was carried out in US in 4 different hospitals under the University of Pennsylvania Health System. Between April and September, they recruited 114 patients with malignancies, either of blood origin or solid organ malignancy, and lab confirmed SARS-CoV-2 infection or presumed COVID-19. And so these 114 patients were recruited, were enrolled into the study, and they found that among all cancer patients, the immune phenotype characterized by low CD4 or low CD8 or CD8 depletion was associated with a high viral load, and this group of patients had the highest risk of mortality at 71%. They saw also, quite interesting, that even if there was impaired B-cell response due to the various chemo regimens that these patients were on, if they had preserved CD8 T cells, then they had a lower viral load and they had a lower risk of death. So again, I think these data highlight the importance of CD8 T cells in modulating the outcome of COVID-19. Okay, now we'll come. This is the last slide for this afternoon's talk. So I hope we have convinced everybody that first, the total binding antibodies and T cells appear sufficient for protection against COVID-19. That's based on our study from the healthcare workers that have received the Pfizer vaccine recently. Then depletion of T cells, but not B cells, is associated with severe COVID-19 in animal model that Eng Eong has showed, and also we saw in the last paper in hematoma malignancy patients. And a lot of these papers that we have shown provided evidence that the early T-cell response was associated with better clinical outcome in these patients. So finally, I think the good news is that we think that vaccine that produces good T-cell immunity will be less impacted by the SARS-CoV-2 variants. Okay, So the last slide, acknowledgement, lots of people to thank for all the work that we have done, and we are happy to take questions after this.
So I'll just kind of pause for a second and say thank you so much. That was a lot of information to absorb. So I'm sure people have a lot of questions, and I can see them coming into the chat group. So I don't know, are they the last few? Maybe we can start from the bottom and kind of scroll up, because the last few have gone back to, I guess, the point that you guys really want to highlight on the T cells. So one question is a very practical question. So which vaccines produce the highest T cells? Very direct. And then we also have, how long does this T cell immunity last? And I'll actually add on my own little bit to that is, what if the T cell response have you seen in naturally occurring COVID infections? So not just the vaccines.
I can start. Yeah, I mean, I think a lot of what Jenny showed just now were Antonio Bertoletti's work on patients. So, although for COVID, it was, the observation was within a relatively short time. There was one part of the study that was on the original SARS patient. That was 17 years ago, and we can still find T cells against them. So to David Vershap's question, I guess, what we know is from the convalescent from an acute infection 17 years ago. We also know from studies on yellow fever vaccine that after 10 years, they could still find T cells against yellow fever. But these were live infection. What we don't know, of course, is how well the memory T cells would develop following an RNA vaccination. That's something that I think we would just have to go on and determine it prospectively. Yeah, so which vaccine produces high T cells? Well, T cells are two kinds. One is called the helper cell, and the other is killer cell, although there's some crossover in terms of the activity. So if just think of it as, one sees something from the outside, one sees something from the inside. So the helper T cells will see the virus that the cells take up and break apart, and then they present all the components to the helper T cells. The killer cells recognize whatever that is made inside the cell. So in other words, the component of the virus must be made inside the cell. And so RNA vaccines and the adenoviral vectored vaccines, they make the spike protein inside the cell, and that's why they make good T cell response. So the inactivated vaccines, like those made by Sinovac, Sinopharm, Bharat, and all that, where they just kill the virus, they'll make good CD4 response, but not so much in the way of killer T cells. You want to add anything, Jenny?
Well, not exactly. I think you have it well covered. In Antonio's paper, I think was the second paper, up to 3 months post-infection, we could still see very good reactive T cells against all the different structural proteins of the SARS-CoV-2. I mean, of course, the real interest, I guess what everyone is asking is how long does it stay? I don't think there's any reason to believe that it will be any different from SARS-CoV-1. So SARS-CoV-1, like we said, even after 17 years, when we tested these survivors' T cells ex vivo, we still saw very good, robust expansion.
What about in naturally occurring infections? What kind of T cell responses have you seen?
I'm not sure what you mean.
So have you looked at, for people who got COVID and kind of gone back and seen, you're looking at the vaccines have given a certain response, but what about the people who've been naturally infected, and have you looked at their responses and what do they still have, what levels?
Yeah, but it's relatively short now, because the disease is still it's at most a year long, right? So if you're asking about long-term immunity, there's no way we can determine. This is something that we'll have to follow prospectively.
So should we go on to some other questions? I'm just kind of scrolling through it. I don't know if any one of them sticks out to you particularly.
Maybe I can take the questions on immune enhancement and all that all at the same time. So there's a question from Minteng on ADE, and then on Jietin on whether you get autoimmune reaction, etc. ADE, the immune enhancement that could be associated with COVID and all that, or is also thought to be associated with coronavirus, is quite different from those that associated with dengue. For dengue, the virus needs to get into those white blood cells, right? And so the antibody offers them a second door into the white blood cells. So what by door I mean, so like we know about the spike protein. The spike protein needs to latch onto this protein called ACE2, which is on our cell. So you think of the spike as a key and the ACE2 as a lock. So the key needs to slot into the lock, turn, and then you get into the door, right? So for dengue, the antibody now becomes a second key to get into the same house, right? So that's the idea of enhancement there. For COVID, that doesn't apply. For SARS-CoV-2, that doesn't apply. What could apply is that sometimes the vaccine doesn't give you a good enough or robust enough protection that it could make disease worse. I say that because back in the 1960s, when the respiratory syncytial virus vaccine was developed and given to children, the vaccinated children go on to develop more severe disease than the unvaccinated children when they got the natural infection, right? And it's not because of ADE, where the antibody enhanced infection, it was that the antibodies made disease worse. The same thing was seen again for SARS and MERS, but in animal models, not in humans, right? So therefore, for COVID, should we be worried? If you look at the commonality between RSV, SARS, and MERS, where you have immune enhancement, the common thread there was that they have very low, these vaccines produced very low killer T cells. And that's why RNA vaccines was very much on everyone's radar screen, because we know how killer T cells would develop, and RNA vaccines will naturally stimulate those killer T cells, right? So those vaccines that don't produce so much in a way of killer T cells, okay, maybe that might be a problem. But actually the subsequent research went on to show that if you have this Th1, Th2, so don't worry too much about that. This is HSA's problem, not our problem. So HSA, when they regulate, when they assess whether a vaccine can be licensed or not, they look at this quite closely, whether the vaccine stimulates this Th1 more than Th2 response. Because actually throughout these incidents like RSV, SARS, and MERS, again, if you have more Th1 than Th2, that seems to be protective, and that's with much lower, it's a Th2 response that's more likely to be associated with immune enhancement. So the short answer to this question, I guess, is that if I know HSA looks at this, and if they approve it, don't worry about it.
Jenny, you have anything to add to that?
Agree.
So maybe I was kind of scrolling through the questions and maybe trying to kind of combine or look at where it's coming from. People also have very practical concerns. So they're concerned, like most of them, although now increasingly they might be single dose vaccines. So far we have the 2 dose. So they're concerned about, how much protection will you get after the first one? Do you need to have a 4-week or an 8-week window? Does that make a big difference? So, in very practical terms, what advice can you give them of the 2 doses, levels of protection, across the different vaccines?
I'll start again.
Go ahead.
We know after 1 dose, as I showed you, right, the curve flattened out. So, technically 1 dose is enough to initiate protection. The problem is that there is no clinical trial data to tell us how long that would last without that 2nd dose, right? So everything that we know now is based on a 2-dose regime. Now, I think what UK is doing, scientifically, I'm agreeable. The problem is that you don't know how well it would work, right? I think the immune system would last much longer than 21 days or 28 days. But exactly how long, that's where the unknown is. And if you follow Twitter and all that, right now, some people are saying, oh, up to 5 weeks is really good protection. Beyond that, maybe it starts to wane a bit, but I haven't seen any data. So we can delay the 2 doses and lengthen the interval, but at a point where we don't know how it's going to impact the efficacy and possibly effectiveness of vaccine. So if possible, I wouldn't delay. Just do it the way it's been tested, and then we know what to expect.
Yeah. So maybe from a more practical point is, I know some people after receiving the first dose of the Pfizer vaccine, because of certain side effects, probably were either asked to delay the second dose or have been told they cannot go for second dose for individuals who say rashed out after the first dose of Pfizer vaccine. Then I suppose the real world data and the phase 3 data suggests that even after one, that Eng Eong says, right, even after one dose, there is already protection. So that's good news. And if there is a different vaccine construct, there's not an RNA-based platform that gets licensed, then I would suggest that I would definitely encourage these individuals who go and get their second dose that's from a different platform. Yeah, but we don't know when that. I don't think we know when we will have an alternative to the mRNA-based vaccine at this point.
And I guess following on that, I see a question about, should infected COVID-19 patients still be vaccinated? I mean, I know there's a lot of literature out there, but since they've asked this, let's address it.
There is no, in fact, there are more, there's more evidence now that, the vaccinated individuals, when given a vaccine, they still mount an immune response. So it's not like when you give the vaccine, nothing happens, right? That something still happens. Now, of course, if you ask from a scientific perspective, do they need it? Do someone who's recovered from COVID-19 still need a vaccine? And then I think the answer is probably no. But in reality, it's going to be very difficult to differentiate the two. You actually then need to administer a test to know whether you have antibodies from a prior SARS-CoV-2 infection, because some people can be asymptomatic. And then after that, or they forget, right? They forget that they've actually had COVID-19 or things like that, right? So in practice, it's very difficult. So if it's safe enough, I'll just give it to everyone that comes forward for vaccination.
Okay. Another question that has occurred a little bit is, what about any evidence on genetic disposition? Has that made any difference?
Yeah, so there are, I guess. In fact, listening to the news this morning, someone is suggesting that the fatality rate tracks almost or is correlated with the rate of obesity in the nation. So the higher the rate of obesity, the higher the death rate from COVID-19. The lower the obesity rate, the lower the and they use Vietnam as the example, where the country has low obesity rate and also low mortality rate from COVID-19. Now, whether that is true, still remains something to be investigated, right? There are others that have been thrown around. In fact, I just reviewed a manuscript that suggested another gene, but I can't say it now. But so there are some suggestions that there are possibly some genetic or possibly some baseline factors that predispose patients to more severe COVID-19. But that doesn't mean that those people won't fare well from vaccination. I think they will still respond quite nicely. In fact, and arguably need the vaccines more than others. But in any case, like I said, if you have to administer a test, then give a vaccine is going to be very difficult.
Right. And maybe some people are genetically predisposed to be protected against it. You know, you gave the example of where they were worse, but maybe they'd be protected against it.
Yeah.
We are seeing, you know, I'm not sure whether this is just anecdotal or just not counted enough, but in South Asia, where people were very scared of, you know, what the impact would be, it seems like it's been a little bit less. And no, there haven't been good explanations of why that's happening. So I don't know if you have any, you know, scientific. All I've heard is very, very much journalistic and anecdotal. But if there's any scientific backing you have, that'd be interesting to hear.
Yeah, yeah. No, I think the you know there's a lot going. A problem with you know, Twitter is that it's a good source of information, but it's also a tough platform to sieve out the true, you know, facts from the noise. And there will be a lot, like, as you rightly pointed out. I mean, again, different countries have their surveillance system have different sensitivities, different specificities, and all that. And so, you know, it's not always easy to compare one country, you know, the number of cases reported in one country with the number of cases reported in another country, right? Yeah, it's. I think we will all need to sit down and take a hard look at the data and know how to interpret them at some point.
Yeah, I think there are a lot of theories and postulations about the kind of lower than expected mortality in South Asia, but certainly nothing very strong in terms of evidence-based medicine that we can point to explain this.
And in the few minutes we have left, maybe let's address the issue of variants. So I see this last question has come in. Would incomplete vaccination of the population lead to a selection for variants that might not be protected by the vaccine? And, you know, I think people have been concerned also of the efficacy of the vaccine against the new variants.
Yeah, I mean, Raihan, good question. Answer is probably yes. So we know that a lot of the variants that are developing right now, like the UK, the South African, all that, is part of the virus's adaptation to humans, right? Because prior to this, their natural hosts are animals, which we still don't know, but now they're crossing over to humans. So there will be adaptation to be more effective in getting into cells through the ACE2 receptor, as well as to escape some of the immune response. And so that's why you see this mutation focusing around the receptor binding domain, so that it binds ACE2 more tightly and then therefore get in. The good news is that, coming back to T cells, is that the T cells don't recognize just the receptor binding domain. It actually recognizes the entire stretch of the spike protein. So for the virus to fully escape T cells, it will need to substantially change the spike. And think about it like changing the key. If you change too much of it, you cannot open the door, right? And so that it is detrimental to the virus. So I think the full escape is from you know, a vaccine that elicits both good antibody as well as T cell response is highly unlikely. But certainly incomplete vaccination, where you then let the the immunity levels wane at a point where there's still a lot of virus, could be a problem, right? So yeah, I mean, I think you're absolutely right. Making sure that the vaccines roll out effectively is important.
And should we, right in our very first actually pre-webinar question, there was this very large question. So let's finish on that note. What made COVID-19 different from past pandemics? Both the persistence and the spread. So that's a very big question, but in the 60 seconds you have left, let's just address that.
Yeah, no, well, I think there are several things. Like, this virus spreads, the characteristics very much like influenza, and influenza spreads very effectively, right? I don't think I may be wrong, but I don't think that the rate of spread is any slower than H1N1 back in 2009 when it emerged from the swine, right? So So it has the same characteristics. Perhaps because it causes asymptomatic infection and mild cases, so we don't remove these people from the community fast enough. Unlike SARS, when you got almost 90%, if not close to 100%, who got SARS infection, all ended up in a hospital. And so control is easy. You control the infection in the hospital, you get rid of SARS, right? Likewise with MERS, right? The infection is very severe, and it doesn't seem like it's that as effective, even as SARS, in going from person to person. So the minute you get a virus with a characteristic like influenza, then it will spread. It will spread very rapidly. Right? But it is not the most. It is not the most infectious virus that we have. The most infectious is still measles. So don't stop vaccinating with measles.
Yes, maybe that's our overall message is don't stop vaccinating, not only on COVID-19, but all across the board. But with that, I see we are exactly at our 3 o'clock time. So thank you both very much for this, vast amount of information you imparted in quite a short period of time. And I hope we did get to most of the questions that people asked. There were a lot, so we tried to kind of combine some of them, but we will have this on recording on our YouTube channel as well as our website. And we are always open for, further questions as long as we can, get some people to answer them. And also, please log in to our next webinar, which will, I think, probably remain connected to the COVID-19 pandemic. We're going to look at the economic implications of it. So a little bit of science and business combined. And with that, thank you again. It was delightful to spend this time with you. And thank you all to the audience for being with us also. And we look forward to seeing everyone again.
Thank you.
Thank you. Thank you.